Next, we will see the gene regulation at processing level. So processing level, it shows how an mRNA, that is pre-mRNA, is being processed to functional mRNA, and how it regulate the process of gene regulation. Okay, the first example is, as we all know, what is alternative splicing means. So alternative splicing is the process in which a single gene can code for many number of proteins. Especially in eukaryote, it is possible that a single gene can code for many polypeptides or proteins by splicing out the exons or introns to new combinations, which result in new proteins. So uh, in some cases, specific exons can be retained, or it determines whether a specific exon should be retained in an mRNA or it should be spliced out. Okay, one example for this is the fibronectin. It is a protein which is present both in blood plasma as well as in the extracellular matrix. So the fibronectin, which is present in the matrix or extracellular matrix, which contain two extra peptides than that of the fibronectin, which is produced by liver cell and is secreted to the plasma. So in the pre-mRNA, these two exons, or the exons which are which code for that two particular two extra peptides, will be excised off when it is in the liver cell, and that will be retained when it is in the extracellular matrix. So how this happens is that there are regions during this alternative splicing. There are regions in these mRNA which are marked as weak points. Okay, they are described as weak, and that weak regions will be sometimes will be bypassed by the splicing machinery, and so that that particular exon or exon will be included in the mRNA. Okay, in this case here we can see there are three exons are there: exon 1, 2 and 3. These three exons, the exon 2, in which the splicing enhancers will bind to these particular exon 2, or the region, it should be bypassed or retained, or these splicing enhancer, the binding of this splicing enhancer to this exon 2 will result in other regulatory proteins to come to and bind to these exon 2. So what happens is that this result in the inclusion of that exon in the pre-mRNA, or in the final mRNA. So that exon will be included, and if that regulatory proteins are not produced in the cell, it cannot bind to that exon, and that exon will be excised out from the mRNA. So it will result in exon 1 and only in exon 1 and 3. Whereas if the regulatory proteins bind to this splicing enhancer, it will retain the exon 2, so that it becomes exon 1, 2 and 3, so that it becomes so that it will result in another protein. So these two proteins are actually different because of the presence and absence of exon 2. So this is how this regulatory or splicing, that is alternative splicing, works. Next, come to the translational level control. So in translational level control, this is the structure of an mRNA, eukaryotic mRNA, and where we can see at the end we can see this 5' end we have the 5-methyl guanosine, and the 3' end we have the poly A-T. So after the 5' end we can see a short region which is known as the UTR, or untranslated region, that is called 5' untranslated region. And after the stop codon, between the stop codon and the poly A tail, we can see another region known as 3' UTR or 3' untranslated region. There are these two untranslated region have got tremendous impact or tremendous effect on significance on the stability of an mRNA. So how much these UTRs are there in an mRNA? It actually increases the stability of an mRNA. So this UTR actually determines the stability of an mRNA. And another important factor is the cytoplasmic localization of mRNA. Here in Drosophila embryo, we can see that there are two kinds of mRNAs are there, which is in the anterior region, the mRNA which are concentrated, which we call bicoid genes. That is the bicoid genes which transcribe to mRNA, and there are on the posterior end there are some genes known as OSCAR genes. These OSCAR genes are transcribed to OSCAR mRNA. And the importance of this bicoid and OSCAR gene is that the bicoid mRNA, which get translated to proteins which are actually needed for the formation of head and other thorax, etc. Whereas the OSCAR mRNA that translated to proteins which actually helps in the formation of germ cells and other abdominal organs, etc. So this is how the cytoplasmic localization of mRNA actually determines the what is called the synthesis of proteins or the target proteins that are synthesized according to their morphology. Next is the control of mRNA translation. The mRNA translation is actually controlled by the translation factors, which we have already seen in the process of translation. So when a cell is under stress or during heat stress or under any infection, these kinases, some kinases will be activated. These kinases actually phosphorylate the EIF2, which is the eukaryotic initiation factor 2, which actually brings the tRNA to this translation machinery. So the phosphorylation of this EIF2 will result in decreased ability of this EIF2 to bring that tRNA to this to the venue of translation, so that it result in decreased translation process. So this is how these translation get controlled by these translation factors. Another important factor or another important feature which comes under this translational control is another example which we can note is the production of ferritin. So ferritin is actually protein which is synthesized when the body has excess amount of iron in it. So when we when we intake more or excess amount of this iron, what happens is that this ferritin bind to the iron so that it neutralizes the effect of toxic effect of excess amount of iron in it. So when small amount of ferritin is present, this IRE or iron response element, this is the particular regulatory area or regulatory portion on the gene. To this IRE or iron response element, and another protein known as IRE or iron response iron IRP or iron regulatory protein will bind. So when small amount of iron is present, this IRE will be bound by IRP or iron regulatory protein, so that no synthesis of it is actually a repressor protein, so that no synthesis of ferritin happens. But when the iron is excess in our body, when the iron is excess in our body, the presence of excess amount of iron will modify the structure or the conformation of the IRP or iron regulatory protein, so that it can no longer bind to the IRE regulatory element, okay, or response element and response element. It can no longer bind to this IRE, so that it leaves that region IRE, so that the gene regulation or gene repression is stopped, so that it transcribed to mRNA, and mRNA get translated to protein, that is the ferritin. So the ferritin which is produced will bind to the excess amount of iron in the body or blood, so that the toxic effect of iron can be reduced. So these are so it comes to a conclusion that is these are the three levels of gene regulation in eukaryote, that is transcription level control, then processing level control, and translational level control.